Good evening, my dear friends. This is a video about the genetic codes. I mean to say the codons. The list of triplet codons are given over here. As you can see, the short forms for the amino acids: PHE is for phenylalanine, LEU for leucine, ILE for isoleucine, MET for methionine, VAL for valine, SER for serine, PRO for proline, THR for threonine, ALA for alanine, TYR for tyrosine, HIS for histidine, GLN, it is written over there itself, glutamine, ASN asparagine, LYS lysine, ASP aspartic acid, GLU glutamic acid, CYS cysteine, TRP tryptophan, ARG arginine, SER serine again, and GLY for glycine. It's a very big list. Now I have some shortcuts for you. For the first section, we have some tricks. I mean to say, if we learn these things, feel your love in my veins. Feel, PHE, phenylalanine. You, your love in my veins. Feel your love. Here we have love. Love means leucine. In my veins. I is for isoleucine. My, M for methionine, and veins, V for valine. This way we have to remember. All the names you have to remember like this. If you know these shortcuts, if you know these mnemonics, you can easily remember this, I hope. Second section is serving protein to 3 alumnis. Similarly, the third one, tires have glitchy ashes lying over asphalt glue. The fourth one is sister's trick by arguing servile urgency. Gosh. There are 20 amino acids that we know that help in polypeptide synthesis. If the codons were taken as doublet codon, each will be made up of 2 nitrogen bases, where 4 to the power 2 makes 4 into 4, that is equal to 16 codons, which are coded for only 16 amino acids. So 4 amino acids are left out. How can we have those? In order to have these 16 amino acids, we can make a chart like this. UCAG and UCAG. We get the 16 amino acids like this. So here we have the 4 amino acids left out. So we did not use this way of codons. The first triplet codon was demonstrated by George Gamow. The experiments on this was done by Nirenberg and Dr. Hargobind Khurana. If the codon is taken as triplet, each codon is made up of 3 nitrogen bases, as we use it nowadays. So it comes to 4 to the power 3, that is 4 into 4 into 4, that makes it 64. Out of this, only 61 codes for amino acids. The other 3 are used as top codons or terminating codons or nonsense codons. Now coming to the stop codons, we have 3 codons known as nonsense codons. Out of that, one is UAA, which is known as ochre. Next one is UAG, which is known as amber. And the third one is UGA, called opal. Now about the characteristics of genetic codons. The first character is all the codons are universal. Starting from bacteria to human beings, all have same genetic codes representing their respective amino acids. Number 2, the codons are commaless. That means no punctuation marks are present in between the codons. They do not have either commas or colons or semicolons or full stop, nothing. Number 3, the start codon is taken as AUG, which also codes for amino acid methionine. In case methionine is not present, GUG can be taken as start codon, which usually codes for valine, but if AUG is not available, it codes for methionine. The fourth one is codons are non ambiguous. It means, in Hindi we mean to say it is vishisht. Each codon codes for a particular amino acid only. Number 5, all codons are degenerate. That means only amino acid may be encoded by more than one codon. Here we have exceptions, that means tryptophan and methionine, they have only one codon each. They do not have more than one codon. Now coming to wobble hypothesis. We have wobbly base or wobble base which is not sthir. वो स्थिर नहीं है. Here, what is wobbly base actually? It is the third base of all codons, all triplets, which are like this. If we have UUU, UUC, UUA and UUG, the third codons in each case is, first one is U, the second one is C, the third one is A and the fourth one is G. With these differences, the amino acids are coded differently. So this kind of different, which is not sthir, are known as wobbly bases. Then we come to mutations. Codons are not affected by are always affected by mutations. For example, in case of sickle cell anemia, the beta helix of hemoglobin normally has GAG as their triplet codon. But if there is a mutation arising in the beta helix, then GAG changes to GUG, which codes for valine. That means in sickle cell anemia, GUG is for valine, and thus mutation affects the codons. So thank you friends. Please like and share my video. Thank you.